Good morning. Guess what? It's another e-learning day. So, we're going to do the other part of the packet that you picked up on Tuesday. If you weren't there for Tuesday, you misplaced your packet. That's fine. There's the note guide. Click on that note guide. It says chapter 11.2 notes. File, make a copy. Okay. Then here's the presentation, just like what we did yesterday. Gonna make a video of this so you can hear me. Make sure that you have all the important stuff written down. And there will be a learning check that you should do after you take these notes. And that will be part of your attendance for the day. Okay, so notes. Intro to genetics. So today we are going to review a couple things that we talked about on Tuesday, and then we're also going to go a little bit beyond what Mendel talked about. We've been talking about Mendel and the pea plant. Okay, so these things we should kind of understand, and so you don't need to write this. Homozygous organisms are true breeding for a particular trait. Uh, like the mask that you see on the huskies. Plants with the same phenotype do not always have the same genotype. Okay, so this is where we have to remember phenotype and genotype. What do we mean? Phenotype does not equal genotype. So, tall tea plants can be heterozygous for being tall or homozygous for being tall. That's what we're talking about. If you have brown hair, I don't know if you have brown hair because both parents gave you a big B or if one of your parents gave you a big B. Okay? The likelihood that something is going to happen is called a probability. And we see this not only in genetics, but also in sports. We have a big sports event coming up. Maybe some of you plan on watching it, the Super Bowl. There is a probability, it's a 50-50 chance. No, there's actually more than a 50-50 chance. I don't know what the probability are that the Bengals are going to win, but I do know the Bengals have lost the last 2 times that they have gone. So we will find out what happens. But let's go back to flipping a quarter or a coin. I flip a quarter. What are the odds I'm going to get to come up with heads? There's only 2 sides. It's always going to be 50-50. The football game, of course, they do a lot more than, it's not just a flipping a quarter, it's comparing the quarterbacks and touchdown rates and everything else. What's the probability if I flip a coin twice in a row it will come up with heads? Does it go up? Does it go down? In other words, does what happened before affect the future events of flipping that coin? No. The coin doesn't know what happened. If I flip the coin once, it comes up heads, I still have a 50% chance that it comes up heads the next time. The same is true of genetic crosses. So just because my first child had brown eyes does not mean that my second child had to have blue eyes. If you remember what we were talking about on Tuesday, how I know my dad had blue eyes, I don't have blue eyes, but my daughter does have blue eyes. My son does not have blue eyes. Okay, so the principle of probability. This is saying that the alleles segregate and come together are totally random. So, which allele you got from your mom and which allele you got from your dad ended up being totally random. Same thing for your sister or your brother. Probabilities do not predict the precise outcome of an individual event. The Bengals upset the Chiefs to make it to the Super Bowl. They weren't supposed to win, but they did. How can I be sure that I get the exact ratio? You have to do it many, many times. Um, if you ever get into breeding animals, you know that you have to breed them a lot of times to verify that you actually have the genetics that you expect. So, the larger number of offspring that you get from a genetic cross, the closer you will get to expected values. I mean, you could randomly get the right time out of a group of four, but that would be totally random. So it will be more likely to match if you have hundreds or thousands of individuals. And Mendel could do that with his pea plants. Okay. I can predict the outcome of multiple genetic crosses by calculating the probability. And here's Bozeman, and he can talk to you more about that. Moving forward to a different idea, the idea of independent assortment. So the idea that whether or not you have blonde hair or brown hair, blue eyes or brown eyes, are independent from each other. You can end up with brown hair and blue eyes, or blue eyes and brown hair and blue eyes, or blonde hair and brown eyes. In this case, Mendel is following two different genes as they pass from one generation to the next. So in his case, we are following pea plants. So he started with two true breeding plants. He had the round yellow peas, double dominant, and wrinkled green peas, double recessive. That makes the F1 generation entirely and completely heterozygous. Okay, so in this, he could not tell that they assorted independently because all of them are round and yellow, because those are the two dominant traits. So he let them cross to each other. Remember what we talked about yesterday? The plants are self fertilized, they are perfect flowers. He really didn't have to do anything, he just left them alone. And he found that he had round and yellow peas and wrinkled and yellow peas, and round and green peas and wrinkled and green peas. This means that they assorted independently from each other. He did not end up with only round and yellow or only wrinkled and green. What was the probability of round yellow peas in the F2 generation? Okay, so we can do this one of two ways. We can do a double Punnett square, or we can work with probabilities, where we figure out what genotypes give us round yellow peas, and we do the math. This is what I was saying the other day, where we can do Punnett squares for the easy things, or we can do the math. So the odds that I get big R or two homozygous dominant or heterozygous dominant. 1/4. Think about the basic Punnett square. If I was only doing the Punnett square for big R for the round, 1/4 will be this and 1/2 will be this. You can do the Punnett square if you don't believe me. So 3/4 of them are going to be round. And if we do the same thing for the yellow, we get that. What are the chances that we get both round and yellow? You multiply across. 3 times 3 is 9. 4 times 4 is 16. So then 9/16. So out of 16 plants, 9 of them would be round and yellow. If I wanted to do a Punnett square to figure these out, I have to remember the alleles segregate independently. So I have to figure out all the different combinations that we can have for both eggs and sperm, which isn't difficult since they're the same. Then when they come down and across, I have to put the letters together alphabetically, and then the capital letters before the lowercase letters. There we go. And then I have to start counting how many are going to be the dominant trait, how many are going to be which trait. So I know that I have all of these phenotypes available. Now I have to start counting the genotypes. And the genotypes. And the genotypes. So out of 16 squares, I get 9 out of 16 are round and yellow, just like I did with the math. 3 out of 16 will be round and green. 3 out of 16 will be wrinkled and yellow. And 1 out of 16 will be wrinkled and green. Now, if you thought that was complicated with just 2 traits, imagine if we add the third trait, tall and short. Well, if we add the color of the flower, then we have 4 traits. So yeah, it gets really complicated if we want to cover multiple genes in a Punnett square. And that is where the math, going back up to a math problem, makes it actually easier. It sounds like it's harder, but if I do each square individually, what are the odds that I will have this? Someone asked me this, what are the odds I'll have round, yellow, tall, and whatever color of pea flowers, I forget the color of the pea flowers. I could do it because I would do each square individually just like that. Then I would take all of those probabilities that I will have this one, this, and multiply it across, and I would have my right answer, rather than having to do a Punnett square with a huge number of squares. Okay, so that was independent assortment, that the different traits would be separate from each other. Then we have beyond Mendel. We have multiple alleles. So we have more than one allele. So before we had tall and short, or green and yellow. In this case, we have blood groups. In humans, you are either A, B, or O. A and B are codominant, meaning that you can be type AB. O is recessive to both. So maybe you are type A, maybe you're type B, maybe you're AB, maybe you're O. 60% of Americans are type O. Positive and negative are a separate gene. So you may be O negative or you may be O positive or some other combination. So A and B and O are 3 alleles for the same gene. Polygenic traits. I mentioned this very briefly Tuesday, that skin color and height are more than one gene. And Bozeman goes into this on how you can kind of understand it. Basically, you can think of the different genes for skin color and height as being kind of almost an on-off switch or a tall short switch for height. And so the taller you are, the more tall genes have been switched on. The shorter you are, the more short genes have been switched on. The more fair your skin is, the more the color of the color gene has been switched off. And the darker your skin tone is, the more the color genes have been switched on. And therefore, you tend to be somewhere in between your parents, but it is possible to be darker than either parent because each parent could randomly end up giving you the darker colors of both. Or you could be taller than both your parents because you ended up with the tall genes from both sides of the family, or the short genes from both sides of the family. Okay, we mentioned in the ABO that A and B are codominant. Co means you see them both. Okay? So here, this chicken, you see both black and white feathers at the same time. They are codominant. It's like having co-teachers. Both teachers are in the room at the same time. Okay, so codominance. Both alleles are expressed and visible at the same time. Notice we are using different capital letters because they are different and both visible at the same time. So if I was going to do an ABO blood group problem, I like to use a little I for O because O is recessive and it's hard to tell a big O from a little o. And since A and B are codominant, you use the capital letters for both. So if I had someone who is A positive and someone who is B positive and they had offspring together, you could get all 4 blood groups if both of them were carriers for the O. You would also get AB and B and A. Then we have incomplete dominance. Incomplete means they're not quite dominant and they end up blending. So sometimes we will see this in flowers. There's a time in rabbits too, but I don't think I should go there. Okay, so in flowers, it's easy to see. I have this red flower, I have this white flower. I end up with pink flowers if I mix them. In this case, they're using superscript. F is for flower, for whatever type of flower this is. And so when I blend them, FR, FW means pink. If I have FW, FW, I have a white flower, and FR, FR is a red flower. If I used the capital letters like I did for the ABO blood group, it would mean that I have red and white flowers. So it's kind of important to do something different, though sometimes people don't do that. But you have to read carefully and know this is an incomplete dominance. So I use a superscript here so you could see that there was a difference. Sex-linked traits also use superscript because only the X chromosome carries the sex-linked trait. The Y chromosome is much, much, much smaller. Okay, so notice there are no alleles on the Y chromosome. And the Y chromosome is what makes the males biologically male. Well, actually, it's a single gene on the Y chromosome, but we'll just go with this. Okay, so H is hemophilia. That is the trait that causes hemophilia. That's the one that causes people to bleed to death if they get a small cut without medical treatment. So if you have a male with hemophilia and a female who's a carrier for hemophilia, you would get females that have hemophilia and males that get hemophilia. Until recently, you would not see this, males with hemophilia, because they would bleed to death before they were old enough to reproduce. If this was color blindness, you'd see a little C there. You'd see a C instead of the H. And color blindness just makes it so you don't see certain colors. Usually red-green color blindness is the most common. The last type of genes that are beyond Mendel, genes in their environment. So there's the Himalayan cat, the Arctic fox. There's also the Himalayan rabbit, or the Californian rabbit, which is actually the same gene, where temperature affects their hair color. There are certain diseases where if you have the allele for that disease, and then you trigger it with the environment, you are guaranteed to get that disease. So there's an emphysema, a severe disease of the lungs, where if you smoke any type of smoke, it will guarantee that you will have emphysema and will probably die of emphysema. The people of the Norway, Sweden, and Denmark are more likely to have that allele. And so they should never ever smoke because they're more likely to carry that. And this is just an example of a incomplete dominance. I have a red and a yellow, and I end up with a orange flower. And I bet that shows up on a quiz or a test somewhere. Okay, that should be all the notes. That you need. And I will have the learning check completed and on the agenda for tomorrow. For Thursday. I'm doing this Wednesday night. So I will talk to you a little bit tomorrow.